more familiar with this technique, while we're going to explore some technique, biosensing technique, and you should know this kind of terminology, like what is non-invasive, what is the invasive.
Hello, sir.
Hello.
Sir, your slide is not in full screen mode. Sir, actually, it is covering some content and it is very small visible.
Yeah, because my computer is connected to another monitor. That's right. Let's disconnect one monitor. Right. It is in full screen mode, but it is connected to another monitor. Okay, let's disconnect. Just mm. Is it possible from here? I think it should. Okay, it's better. Is it okay?
Yes, sir. Yes, sir.
It's fine, right?
Yes, sir.
Hello?
Yes, sir.
Good. Okay, let's start. See, non-invasive means it is kind. Do you know the definition of the non-invasive? Someone, can you please? Anyone, can you please tell me what is the non-invasive? Some definition. I think you may know, right? Do you know anyone? Can you please tell me?
So reusable.
No.
Sir, which is not penetrable to the body.
Means see, when you're going for diagnosis, right? Doctor using, maybe they want to test your blood samples. So how they're collecting the samples? They're pricking your finger for like glucose test, or maybe they can insert a needle, right, and take some blood samples. So it's kind of painful, right? That is called. I mean, sorry. That is called invasive method. Invasive means that is the painful method, like pricking the finger or using the needle. That is the invasive. So, what is the drawback of the invasive? You can think about, like, the common example is the diabetes, right? So, generally, you have to you should know the level of glucose before meal, like before the lunch, before dinner. You should know the level of glucose. Especially diabetic persons, that's why every day you don't know how many times they're pricking their finger for testing the glucose. So it's very painful for them, really. Sometimes they become the panic. And another thing, especially, just think about some like children case. Some developed countries nowadays, like very early age, within like 2 years, within 5 to 10 years, they have also diabetes. Just think about, also not only that, like like baby, like 1 years. So that case, you just imagine how many times you can prick their finger. So that's why some alternative method we have to think for testing. Like, if we can, you can see the image number 1. Just we can develop a sensor, like printed sensor on a like thin film. Just see, on a thin film we can print our working electrode, counter electrode, reference electrode. On the working electrode, we can fabricate our expected all the chemicals or others, bioreceptor, anything that your sensing material, everything will be on your working electrode. Then you can touch your tongue on the working electrode area. So what will happen? From the saliva sample, they can detect, basically. So it is fully kind of the non-invasive. So non-invasive means it is kind of not painful technique, and without pricking finger or using the needle, you can test your. You can use your biosensor device. So it can be wearable. The main very important thing, very fundamental things you can use here, like the wearable sensor, you can develop here by using this principle, right? Nowadays, like the bandit type, you can use on your screen, and from the sweat sample, they can detect different kind of the analyte. So something like wearable sensor you can develop. And so this kind of sensor you can form basically very small, very small sensor device, and definitely they will be portable. So it can fulfill that, I mean, the point of care, that technology, that all the requirement it can fulfill. That's the method I'm telling here. So you can use like wearable kind of thing for non-invasive. You can see in the right hand side on your screen also you can print your sensing material. All the bioreagent can be printed on your screen here. And it can absorb the sweat sample, and from the sweat it can send the data to the wearable device, or maybe some mobile device you can use. Some digitally you can see the value. In the middle picture, you can see here, there is another technique we can use. Sometimes, maybe from the sweat or from the saliva, your analyte concentration not sufficient. So what you will do, you can use some very small needle that is called the micro needle. So especially, you can see this hill type. This is called a hydrogel micro needle, a very small needle. But you can say why then, as you are using needle, then how it becomes the non-invasive. You have to develop some kind of the needle that can penetrate your just upper part of your skin, and it will not hurt your nervous system. So you will not feel the pain. Just they will insert, they will penetrate your skin, and just below the skin there is some interstitial fluid. So interstitial fluid will absorb. Then there is, you can see some micro channel. So that fluid will go through this, I mean, from the needle, then through some microfluidic layer or microfluidic channel will be there. So it will come to the working electrode here. Like there will be some counter electrode, reference electrode. They all will be printed on a maybe flexible substrate. So see here in the middle side, so this is kind of a flexible substrate. So we can print all the electrode like working counter reference. Another end, we can use a needle that is called like some soft micro needle. They will not hurt your nervous system, but they can absorb the sample. Okay, like interstitial fluid. And they will send, because you need some amount of the fluid. So there is so many, you can see the channel in the needle, and they all will collect the sample, and they will gather the sample, and they flow through the electrode. And excess amount will be stored in the waste chamber, something like this. You can see the left hand side, it is the full like a banded type food sensor device. You can attach on the skin like a baby. On their skin, they can attach this one, and no need to prick their finger. So whole day they can monitor the glucose. How they will monitor the whole day? So they will absorb the glucose, sorry, I mean interstitial fluid. So interstitial fluids contain the glucose. Then it will flow through this sensor, it will measure the glucose, send the data to the electronic device, because we can connect this one via Bluetooth to your like smartphone. To your smartphone, it can be connected. They will send the data. A whole day, it will absorb the fluid sample, and it will detect the glucose level and send the data to your portable device like smartphone. So this kind of wearable, non-invasive approach is very, very smart technique nowadays. People prefer it. Also, I think you already know some smartwatch. Like Apple already developed some smartwatch also came in the market. So there is a concept similar like this. They can detect the glucose level, and they can monitor like whole day. They can monitor. They can send the data. But some smartwatch, they are very expensive. Basically, this is the drawback. Some smartwatch very expensive, almost like 10,000 rupees also some smartwatch. So that's why, so if you think about like developing countries or rural area, so they will not buy this kind of expensive instrument, right? So that's why one area is open to you. So if you can develop some glucose sensor, non-invasive way, also very cheap, that will be very much demanding technique. So I mean, you can easily bring this kind of technique in the market. So some area is open still. And some wearable sensor, that's I mentioned that for
Sir?
basically we can use. Tell me.
Sir, this kind of skin patches require some potential, right, sir?
Yeah, that's see in the skin patches. This is kind of sensors that or like I already teach you like printed circuit board, right? So on the front side, we can immobilize our sensor, and back side we can immobilize some electric circuit. There actually we can fabricate or allow Bluetooth device everything. So through our portable electronic device like smartphone, you can apply some potential. You can send the command. So that potential whole day they will apply and send the data to your device. So we will use here Bluetooth technology. Okay.
Okay, sir.
Definitely. I mean, sensing technology, whatever I taught you, that technology you have to use. So just I am teaching you some next phase. In other technology is same, like application potential, everything same. Just some approaches I am teaching. Like here, see.
And these are time durations we can also modify.
Time duration.
After
What do you
what
mean?
time, sir? After what time it will going to give us the readings? Means we can.
Yeah. That actually we can optimize. See, like maybe with like every 1 hour you need the glucose level, right? So you can customize, you can run a program, everything. So that I think that very common things it is in your hand. That programming things that you can do by yourself, like that program you can write and you can load basically. Every 5 hours, not only every 5 hours, maybe you wanted to know the glucose level at 12 o'clock, at 5 o'clock, at 9 o'clock. So something like this. You can customize. Okay. So
Okay, sir.
that is the that technology I think already developed. People know how to do this. Like who are from the electrical engineering background, they know very easily. I think also we can program our portable electronic device. The cyclic voltammetry, chronocoulometry, chronoamperometry. How much potential you are going to apply, how you can convert the current amount to the digital value. Everything can be done. I think basically for this we need to collaborate with the electronic engineer. They can do this easily, and you become the sensor expert. When you become the sensor expert, you can fabricate the sensing part. So that's why I'm saying this is very much interdisciplinary area. So if you are from the electrical engineering background, you can cooperate in this part, like how we can fabricate the electronics part, like Bluetooth part, like programming part, these things. And now, although you are the electrical engineer, but you are learning the sensing technology, right? So now you become the complete person who knows the technology, basically, whole technology, right? So you know how to fabricate the sensor, you know the electrical circuit design, you know the program. So you become the complete, right? Clear? Okay.
Sir. Yes, sir.
See here another thing, like it is. I think this is not new, but still this research going on. This is this kind of wearable sensor. Do you know what is this? I'm showing this cursor. This sensor we can use from the tears. You can detect the
Price. Price.
Yeah, so you can from tears also, so if you search, if just Google it, from the tear there's so many analyte you can detect. Tear sensor, I can show you in the Google, just you can see here, tears. The biosensor.
Sir,
Hello.
are there any sensors, biosensors related to saliva?
Definitely. There is so many sensors already developed from the saliva, like not only glucose, you can develop. So if any, like see, malaria, some Plasmodium falciparum PFHRP2, that is also available in the saliva sample. So you can detect basically different kind of the malaria. Not only that, right, like the SARS-CoV-2, like COVID-19. This is also available in the saliva sample. So any kind of analyte target, if it is available in the saliva, so for them you can develop. Okay, and collecting the saliva sample is very easy. It's clear now. Hello.
Yes, sir, it's clear, sir. You were searching for TS.
Now, see, yeah, you can see if you see paper-based, carbometric-based tears, glucose. Yeah, there is so many. The recent advancement portable. See, I can download on paper. So from the tears also, that I am going to show you today. That not only the blood samples, there is so many other samples also we can focus on. And non-invasive way and very smart device basically. If you think about this kind of the sample, so very smart way you can develop some sensor device. Like if you can develop some, people are wearing the now contact lens, right? So on the contact lens you can fabricate some small chip, sensor chip, that already shown here, right here. That small chip you can fabricate. So I mean, then whole day they can detect the, whatever you want, like glucose also if you want, or some other thing. So your first job will be, you have to check the level of that analyte in the sample. So if you're using like saliva sample, but in the saliva sample, if your analyte concentration not that much high, then it is very difficult to detect that analyte by using your sensor. There is another way. If it is very low, then you have to develop some ultra sensitive biosensor. Means its signal to background ratio should be very, very high. That you have to develop. That is one thing. So you have to check the level of the analyte in that sample first. If you are going to use the tear sample, then you have to check like glucose level, how much it is in the tear sample. So something like this. So you can see here the recent advances in the portable biosensor for biometrics in the body fluids. So many kinds of body fluids are available. So you can check here. So like this way you can search. You will get the many samples, many techniques that they are using for the detection of biosensors. You can see here the electronic picture also here. Okay. See, so this is also another wearable sensor you can see. So this is a flexible. So maximum wearable sensor are very much flexible, like this. So they can wear in their arm, and during the exercise they can measure the body, the glucose level or whatever. It depends on the sensor design. Something like this. So this kind of the sensor also you can think. Okay, so you can search like this. Okay, let's come back. See, in this in the right hand side, this picture. So this is also, right, I think. See here. Generally, this is the smart watch. On the right hand side, you can see they can measure your something like glucose, or not only glucose. As I said, any kind of analyte you can measure similar way, and they can send the data to the your mobile phone via Bluetooth. You can see the digital value. So this kind of technology is very, very smart. So people are looking for to develop some sensing device using this Bluetooth technology, and like this you're using your smartphone, because maximum user nowadays people are using the smartphone. So if you can generate some sensor and you can integrate you easily with your smartphone, that will be very, very advanced, and easily you can bring them to the market. And developing a startup, these things you can imagine like this way. Okay, clear? Yeah, so this is the advanced technology that I mentioned. Let's come some new technology that is called some environmental monitoring. That is another topic actually that I wanted to cover in your syllabus. So slowly I'm going to complete your syllabus. So some new things actually I'm going to teach you. So another thing is the environmental field monitoring. Food safety, security. These three points is very, very important. So why I am looking for the environmental field monitoring? So nowadays we are knowingly or unknowingly we are polluting our environment, right? Or we already polluted, but now we become very serious. But so you should know the level of your pollutant in the field, like the water sample in your river, or like the maybe soil sample in your agriculture field, right? Maybe your agriculture field you use lots of fertilizer, too much fertilizer you already use, right? So what happened then? It become the nitro. I mean, because of the fertilizer is mainly the nitrogenous fertilizer people are using for getting the more and more and more productions. So they're actually basically polluting their soil. And another problem, so when you're using more and more fertilizer, means nitrogen, so that part can come to your drinking water also. So from there, one I think blue baby syndrome, maybe you heard maybe. This is because of this nitrogenous issue. So there's so many side effects for that. It can affect your nervous systems. So growth can be hampered. Your mental growth can be hampered. So this kind of problems should be solved now urgently. So we should develop some sensor who can which can measure the pollutant in the environment, like soil sample or like in the water sample. So arsenic, this kind of pollutant also is well studied already, but still you can develop some and like from the river water. You can dip in the river water, you can measure the arsenic level, or you can measure the nitrogen nitrate level, basically in the form of nitrate in the fertilizer we are using. So that nitrate level we can measure, or phosphate level we can measure. Also lead, another contaminant in the water. So we can detect the lead level, or something like this. Then another point is the food. So sometimes food become the poisonous. They already spoil like milk, right? So in the like people sometimes in the store room they're storing the milk in a long time. They may not be good for your health. So you have to taste that sample how. And for that maybe we can develop some sensor. So many food like many fruits available in the supermarket, maybe they are not eatable, or maybe their quality not that much good for your health, right guys? Have you think about this kind of thing? Have you thought like this?
Sir. Yes, sir.
Right. Hello.
Yes, sir. Yes, sir. We have thought about it.
Yeah. So for that you can develop some sensor, right? So actually I am telling you the area where you can explore. I'm just showing you the area where you can learn, you can search papers, you can get more knowledge, and based on that you can maybe do some project on your masters, or also you can do your work during your PhD, and you can develop some new sensor. So this is the possibilities basically I am telling you. So food safety is very, very important. You can think more. Also another thing is the security. So why I'm mentioning this part, security. Security come from this is a terrorism kind of the attack. I think you already heard about this. Like the virus terrorism, or that also possible. So like they can spread some kind of the very dangerous virus, or not only the virus, different kind of the bacteria that can destroy. I mean, this is the very dangerous weapon, I think, in the society in this modern area, modern time, right? I think this also already came in your mind, right? This kind of the national security from the bioterrorism attack. Have you heard this one? Right?
Yes, sir. Yeah.
Yes,
Hmm?
sir.
We have
Oh,
also
so
seen them.
yeah. So for that also, we can develop some sensor. So is there any kind of sample is already affected or not? So if it is affected, then we have to immediately stop. So if you want to immediately stop the spreading, when you have to, for the awareness, everything. So you should have some sensor, right? So that they can tell you that, yeah, this maybe this river water already affected, right? So that immediately you can take the action. This maybe soil sample or any kind of food maybe somehow contaminated, so that we can detect. So the lab on a chip technology, we already use the lab on a chip technology. You know the lab on a chip technology, right? So that concept now we are going to apply. First come to the environmental monitoring, first part. So it is interesting, guys.
Yes, sir.
See, we can say soil on a chip. Last time I taught you lab on a chip. Now we are going the specific talk. Soil on a chip. I mean, it is something kind of on your, you see, you are the expert who are doing the research in this field. So you can put some name. Like if you want to detect, I mean, some disease based on your organ, something like this, you can say organ on a chip. You are going to monitor some disease on your brain, right? So you can say brain on a chip. So something like that. You can change the terminology. So here we are going to detect different kind of analyte in soil. So we are saying soil on a chip means everything can be done on a chip from the soil sample. So this is something like this kind of the representation. So on the lab on a chip technology, generally we are using some microfluidic technology. So we can one end you can drop your sample, then it will be some. Then you can use some maybe buffer, some liquid. You can drop it, and then all the analyte will be dissolved. Then it will go through the. You can see go through the microfluidic channel. It will come to like some. Then it will come to the detection area. Then it will detect the analyte and send the data to your mobile device. So something like this. So let's come here. That I already mentioned that soil problem, like fertilizer. The main problem is the fertilizer. So here I actually so mainly we need the fertilizer to boost the agriculture productivity. So what people they are doing? Add more and more and more. So the farmer thinking if little is good, then why not add the more fertilizer? Then we'll get the more product, right? People are thinking like this, right? Right?
Yes, sir.
Yes.
You know, in the crop field, agriculture field, farmer they many. I mean, I'm not saying that everyone, but many people they don't know how much they want. They should add. Huh? So that they're doing some arbitrary amount like that. They know that if they like 5 kg, if they add like seed in the few area of the field. Then this year they got the very good products. They said, let's add the 10 kg. Then we'll get the double production. But they don't know how they're affecting the environment. So this is the problem. See. So there is main problem is from the fertilizer. Generally, fertilizer contain nitrate, phosphate. Also, we are using the pesticide. You know the pesticide. Pesticide like herbicide. They're killing the basically there are many kinds of the pest like you know the pest, right? Or different kind of the insect. So they are actually basically destroying your production or the crop production. So for that we need some. They are basically some chemical. They all are the chemical. So they have very bad side effect in the human health. You can see they kill the livestock, human health, their hazard. There are so many other problems: cancer, diabetes, thyroid problem. neurologically shoulder can come. So we have to be serious now. This is the time. Yeah, we have to stop using too much like a fertilizer, pesticide, this kind of thing. So how we can, I mean, you can tell them. So how we can say, so you can develop some device, you can, in front of them, you can test that, see, this is the amount already, this high amount nitrate already present. So you cannot use this much. So some awareness you can spread like this way. Yeah. So, you know, right now, so if you can develop this kind of the device, it has huge demand in the market. Portable, this kind of the device, and almost we don't have. So we have to stop using too much fertilizer, pesticide, something like this. So we have to develop. Like here you can see one lab-on-a-chip sensor like this. See, this is very portable, very small. So REM reference electrode, working electrode, counter electrode, we can fabricate. So there is two tubes. So from one tube you can send the samples. I mean, you can dissolve your soil in a liquid, and then you can pass through your working electrode. And on the working electrode, maybe you can develop some enzymatic method or non-enzymatic method. So in this work, people they actually develop some enzymatic, like NIR enzyme, nitrate reductase enzyme. This is called. So if the sample contain nitrate, then it will react with this enzyme, and it will, I mean, you will get the current change. So this kind of very simple, I mean, all the basic sensor technology I already taught. Now the application here. So here, see here they use some like graphene oxide, something like this. On that surface, they use the enzyme. How you can conjugate the enzyme and on the surface of the nanoparticle or graphene oxide, you already know. I already taught you, right? So like this way, very simple technology they use on this sensor, and they can detect. Like you can pass the sample on this area, and you will get the amount of the nitrate. And but there is one drawback. See here, actually they use the nitrate reductase. So this is enzyme, right, guys?
Yes, sir.
So enzyme is this is it can be denatured easily, right? Protein type of things, right? So this is the problem. So there is another avenue or area is open still. I mean, you can explore without using any enzyme, maybe. So they already tried enzyme, they got good result, but there is some problem. There is some drawback there. You can use, I mean, without enzyme, maybe you can try some non-enzyme material, like some nanomaterial you can try here, so that they can, I mean, a nitrate can react on that nanomaterial, and you will get the current change, right? So this will be a very good method, right, guys? Right?
Yes, sir.
Yes, sir.
Yeah. So there is another area still open. Where you can. So whenever you will try some sensing technology, just keep in mind at the early stage, maybe you can try using some biological material, because they are easily available and they are very, very specific. This is the very important point. They are very specific. But development of non-enzymatic method is not easy, but it is not difficult also. You can develop. So what you can do, you can develop some nanomaterial, some specific nanomaterial. You can synthesize them, and you have to test them that it is reacting with your nitrate or not. So if it is reacting with the nitrate, is it reacting with other? I mean, see, in your sample, maybe nitrate present, also maybe some other species also present. But if all together they react with your nanomaterial, then it is very difficult to detect the nitrate level, right? Maybe you will get the higher signal, but that is not only the nitrate, maybe only for the other interfering species also they can hamper your data. So this is called the specificity, right? Your nanomaterial should be very, very specific, right? This is very important point, guys. I think you already heard about this from my side already many times, and you have to consider this point always. Okay. Clear? Clear, guys?
Yes, sir. Yes, sir.
So when you are developing some non-enzymatic method, you always have to consider the specificity. Okay. So although you are developing some nanomaterial, yes, I got some sensor. So it is very stable. There is no enzyme, nothing. But if it is not specific for that analyte, then it becomes useless also. Okay. See here, some paper is.
Sir,
Tell me.
sir, what is microfluidic technology, sir? Can you define it?
Microfluidic technology.
Yes, sir.
See, microfluidic technology means we can use some very I know the micro level. It means milli than micro. I mean very small channel we can use. Why we need the very small and very thin channel? Because we can use very small volume of the sample. Also, because of the you can say that microfluidic, I mean there that is called you can say the microfluidic transport one maker. I think that what is your background? Can you tell me first?
Sir, biotechnology.
Okay, so biotechnology means you don't know the this hydrodynamic method. So I can tell you very simple way. So microfluidic technology means something like very thin channel we can use because of the you see like just give a small simple example like a paper. So if you drop one water on one end of the paper, so what happened then? Can you explain? Can you tell me? If you drop one drop of the water on the paper, sir, paper, what will happen?
So it will spread with capillary action.
Yes, that capillary action. So in the microfluidic channel, that capillary action actually react. So because of the actually paper actually contain this kind of the very thin needle type of things. That is so microfluidic channel we can easily fabricate on a substrate. Okay. So if you drop your sample like 5 microliter, 10 microliter like this. Okay. So you can easily spray it. Also, another benefit, I mean, easily it can go from your inlet.
Sir, it's how it's related to sensor, sir, by sensor.
You can relate with the sensor because if you integrate your microfluidics, this channel with your let's come back. Yeah, see.
All the POCs work by this principle, right?
See, here your sensor working counter reference. Okay.
Sir,
Hello. Hello.
sir, all the chip on paper stuff work on this principle, right?
I'm not getting your voice. Not clear.
Sorry, sir, I'm saying that all the chip on paper, like.
Yeah, all the lab on a chip.
No, sir, like you said, right? Laboratory on chip, yeah, these are all work on the same principle, right? Microfluids.
Yes, we can use the same principle, not like the 100% all. Maybe I think I already taught you some like a malaria detection. Can you remember that was free? Just drop it will come through your paper, then come to your working electrode area. There is no microfluidic channel extra we fabricate, but that is also laboratory. So it is not like every time you have to use the microfluidics. It may or may not. But
Okay, sir.
time people are using the microfluidic channel. Why? See, if you use the microfluidic channel, another point. So if you use the microfluidic channel, you don't need the large volume of the sample. Not also sample, the reagent. You don't need the lots of the, not too much amount of the reagent. You can use very small amount of the reagent, very small amount of the sample, because this channel you can see very small, very thin channel, right? So for that only maybe 5 to 10 microliter if you drop, easily it can come to your sensor area. So why microfluidic channel is related? See, like this part, it can be touched to your skin, right? So slowly it will absorb all the sample, like the interstitial fluid or your sweat sample, then it will go through some channel, right? So microfluidic channel will help in this case. So microfluidic channel will help to flow all of your sweat or interstitial fluid to your sensor area. So like this way, we'll integrate your microfluidics and sensor together. So if there is no microfluidics, how this all sample will go to your sensor area? So not only this, if you don't have microfluidic, you need lots of, around maybe 5 ml or maybe 5 ml, you need some sample. But in this case, it can be done within maybe 10 to 20 microliter or 30 microliter is enough, because they can easily go to your very miniaturized your sensor. There we are developing very small sensor size in very small size sensor. So this channel will help all the fluid to the sensor area. So this is the two main point where we are using microfluidics. So first, we can avoid to use the lots of sample, like amount of the sample or amount of the reagent. We need very small amount. And another thing, we can easily transport. We can easily transport your sample to one end to your sensor area. Okay, clear?
Sir, normally what kind of material are used for this microfluidic channel formation?
How many? Sorry?
What kind of materials we are using for making this type of channel, sir?
Oh, so see, it's depend. That is good question. It's depend. So sometimes we are using like PDMS or PMA. That is a very flexible plastic kind of things. Also, you can try some PCB material, like printed circuit board. That is, they have some their own material, like FR4 material. That also can do. Generally, people are using the like PDMS or PMMA. This kind of the material generally people are using. Many flexible material they are using. So not only this, so people are trying nowadays on the glass also they can easily fabricate the microfluidics. So it's depend on which material you are trying. So you can use like lithographic technique or some. So there is so many other different techniques also available. Mainly the lithographic technique, very old established technique available. So you can pattern your microfluidics channel. Then you can use your photolithography, and you can easily fabricate your microfluidic layer on the sensor surface. So like this. So like this, you can easily fabricate like this kind of a channel. So I will show you also. So this is the technique.
Sir, these are all the same sequences that you are taught in computational pathology, right? Same examples
Yes,
like
yes.
printed.
Yeah. Same, this is kind of the things you already taught some other courses. This
Yes.
kind of. Okay, this is very interesting and novel technology. I think, yeah. So this is also kind of, this is also, you can say, the microfluidic, but this is not so thin. Also, almost, you are flowing the liquid sample going through your working electrode, and then there is another outlet there. But it is not so-called microfluidics because it is not in a very thin channel. So generally, you need some channel to pass your sample through your electrode, and we can that's how we are generally integrate your microfluidic, your sensor like this. So the main things that's I'm repeating again. The main things, you can avoid the volume of the sample, amount of the reagent, and another thing, you can transport your sample easily to your sensor. Clear now? Okay.
Yes, sir.
Okay, see now here some paper based. Some here maybe some colorimetric change, color change you can see. With some reagent, they can react with some your target. Like if there is some ACAG, this is called acetylcholine esterase. So if you want, they are kind of the inhibitor. So if it is there, something like, see, in the left hand side, you can see this one organic molecule. So if it react with some chemical, they can show maybe some indigo color. So this kind of the color change, see, I'm just giving some general idea. See, this is like a paper strip, right? On a paper, one end, maybe you can drop some chemical, right? Now, in a water sample, maybe it contains some nitrate or maybe some pesticide. So what you can do, so in a water sample, you can see here in my cursor, you can see in a water sample, if it contains some contaminant, so you can dip your paper strip, right? So then what will happen, because of this capillary action, that microfluidic type capillary action, you know the paper, if you dip one end, it can easily, water can go to the other end, right? Because of this capillary action of this means fiber kind of things, it's made by fiber. So easily water can transfer to one end to another end. So what will happen, if it contains some contaminant, so it can react with some reagent that you can drop here and it can show some color. Now, if you have high concentration of your analyte that you are going to detect, then it will show the intense color. If you have the low amount of the analyte, then it will show the lower intense color. So you see, means in this work something different. When I am showing I'm teaching you the principle. Maybe some chemical can react with your analyte and it can show some color. Okay, so that reagent you can put one area, means like this circle area you can put here, and you can keep it dry form. Now you can dip this one in a solution that contains the pollutant. So when you dip, that pollutant can come and react with this reagent, it will show some color. Now, based on the color intensity, you can determine the amount of the pollutant. So how? So we can wait here. We can use some smartphone. You can capture the color through using your camera. You can capture the intensity, and it is already well established. Some algorithm and app already available. So you can correlate that color intensity using your app, and you can determine how much pollutant present in the water sample. Okay, guys, it is the main principle you can try. So first, what will be your first job? You means I'm not giving you very specific example here. I'm just telling you the technology that you can think about. Maybe some chemical, they can react with your pollutant that present in the water. It can be arsenic, it can be lead, it can be nitrate. So that means that is one research area is open. You can try. So maybe suppose if it is the arsenic, maybe arsenic can react with some chemical. So you have to find out that chemical. This is the research. And it will show some yellow color. Just think about it. Now, so from that yellow color, the intensity of the color, you can develop some app, and because from this app, I mean, you know, all the smartphone, they can capture the image, and from image we can analyze. So you can use the algorithm, you can use some smart app, and you can analyze the intensity of the image, and we can say how much contaminant present in the sample. Okay. So no need to remember the very specific things. Just try to understand the technology. Okay, guys, clear? Clear?
Yes, sir.
Yes, sir. Yes.
Now come to this example. See, here they use the indoxyl acetate. So this is some chemical name because they already done the work. So your job will be find out some another chemical name. So this is for some acetylcholinesterase for this. So what they did? So this chemical. So one point can come is stability. So although you are dropping your chemical on the paper, maybe that can be easily detached. So you can maybe make some technology how you can keep that reagent on the paper in a stable form. So they use some just a sol gel silica. So silica can hold the chemical long time on your paper. So this is the things that the people are trying. How there is so many problem can come while you are developing some sensor device. So many problem can come. Maybe stability one of the problem. So they just try to fulfill this point how they can overcome the stability. Yeah. Okay, so same thing, like they collect the sample, they analyze by using their smartphone, and then they load this data to the web portal so that people, when they search in the Google, they can find out, okay, in this area this water already contaminated with arsenic, something like that. Huh? Okay, so not only this is called the inorganic contaminant like arsenic, lead, nitrate. So they are the inorganic contaminant. So there is lots of organic contaminant also there. So we some bacteria like the Escherichia coli, E. coli. This one E. coli. Yeah, E. coli. Do you know, guys, what is the E. coli?
Yes, sir.
Yes, sir.
Yes, sir.
It caused the.
Sir, it's present in.
Tell me.
Sir, it's present in our elementary canal.
Stomach also, sir. Human body stomach.
Yes. Also, it can. What it can cause? Food poison. So E. coli also cause the food poisoning. Water contamination. I think some of you from the biotechnology. So I think I can ask you. So who are from the biotechnology here?
Myself, sir.
Yeah. So I think you already heard about the poisoning of food or water by E. coli, right? Do you know?
Yes, sir.
Yes. So I was now letting. I just wanted to say this one to all of you that if you find that some area of the water is contaminated with this, so you have to spread this awareness to the people that don't drink this water, right? So for that you can develop some sensor chip for detection of the E. coli. Okay, so I already wrote some history here. You can read here. It causes a very serious illness, like diarrhea, something like this. Also, kidney can be failed. See, so E. coli can be detected like our sandwich method on the ITO electrode. So you can see the left hand side on the ITO, like avidin biotin method, we can immobilize a primary antibody. So after that, you can drop your water sample. If the water contain E. coli, then it will bind on the antibody. Then you can use another antibody, secondary antibody that conjugated with some enzyme, that is ALP. ALP is the alkaline phosphate that can react with the substrate. Now we're slowly coming in the right hand side. That is our ECC redox cycling. See, right hand side to amplify the signal. Right? Can you remember this one? Right hand side. See here in my cursor. Can you see?
Yes, sir.
It is ECC redox cycling, right? So we are developing E. coli detection on a sensor chip. So we can immobilize E. coli specific antibody on the surface by using avidin biotin interaction. Now, if you have E. coli on your sample, it will bind on your chip. Now you can add a secondary antibody that conjugates the ALP. ALP means some tag that will help you. I mean, you can use like your ECC redox cycling by using some ruthenium complex and T cell. So this is the you see the hydroquinone. This one can come from this substrate that can react with the ALP. So that I already taught you, right? How you can use this kind of the ECC redox cycling reaction, how you can choose the chemical, what will be the starting reagent. So now we are going to use here. This is the practical application of your ECC redox cycling. So you can try to detect the E. coli detection like this way. So you can use your ECC redox cycling, and you can detect very low level E. coli. Means if your river water is contaminated with this bacteria, so maybe there is very small amount of bacteria present, but that also you can detect by using this redox cycling based biosensor. You can see in the right hand side we use the chronocoulometry, and you can see CFU per ml. This is the colony formation unit. This is the something unit that is used for the bacteria. So just know this is the unit for the bacteria detection, CFU. See here 10 to the 3, 10 to the 4, 10 to the 5 minutes, their amount, number of the bacteria increasing, and you can see that chronoculogram, that chronocoulometric charge is increasing by increasing the concentration of the E. coli, and we made the calibration curve, right? So like this way you can detect some bacteria also for environmental monitoring. Also I already told you the food monitoring, like milk quality. So how we can detect the milk quality? Some milk already spoiled, they're not good for your health. So that also you can detect. Means you can develop some biosensor for milk quality to determine. So see in the left hand side you can see here like the biosensor surface. They use some chemical. This one just for the fabrication of the antibody. In these steps you can use. I already taught some procedure here. Actually they use some chemical like this, and for modifications of the carboxylic acid on this surface, then you UDC NHS. Means this is the just one process you can use that I taught already in the very first class. You can use that kind of technology also for modification of antibody, fabrication of antibody on your surface. So this antibody is very specific for some you can say like ALP kind of things that amount will be increased on your milk if it quality decrease. So we can use the anti-ALP, some antibody on your surface. So this antibody is called anti-ALP. So ALP we can detect in the milk sample. Means if it is a high concentration, the number of the ALP will be high on your cheek. So if it is high ALP present, means you can say this milk is not drinkable. So like this, you can identify the quality of the milk. So here what they did, they use some chemical like VCIP, so they can react with this ALP and it will form some color change. It will react with this and it will form this product. No need to remember the name, just you can use this kind of a chemical and it can form this product if this target ALP present. So your sensor surface already contain this ALP antibody. Now you drop your milk sample on your sensor, right? So if your milk sample contained this ALP, so it will bind, and your sensor surface already contains this reagent. So you will see some color change on your paper strip. So basically you are using some paper for detection, sorry, to get the quality of the milk. So you are just using a paper, and your paper already modified with some antibody. So this antibody can capture ALP that kind from the milk. So if your milk quality not good enough, then this ALP amount will be very high. And if this ALP high, then you will get this product. I mean down what you can see that from this CIP to this indigo color product, you will get high and high. So you will get the intense color. So that color now you can take a picture from your smartphone, and using the app you can analyze this image and you can say how much target present, how much ALP present in your milk sample, and you can guide the user that should you drink this milk or not. Clear, guys? Okay.
Yes, sir.
Yeah. See, I already taught you all the basic principle, right? Basic technology, basic fabrication. Now just I am showing you the application part. Some paper-based chip, how you can develop a paper-based sensor. So something like this paper-based sensor you can develop. So on the paper, you can immobilize the antibody. You can just drop in one drop of milk. Then take the image. You can analyze the image, and you can say how much target present. So here you can just see this is the calibration curve we made, and and as I mentioned that your sensor should be specific. So if you want to detect the ALP, means then other interfering species they should not hamper the data. See here is citric acid, BSA, lactose, other also present, but they are not hampering your signal. But if you have ALP, then only you are getting the signal. So this is called the specificity test. Clear?
Yes.
Yes, sir.
I think some of you are already very silent. I'm not getting. Let's.
Sir.
Divanshu Gupta. Divanshu. Hello. Yeah, someone wanted to ask me something. Yeah, tell me.
Yes, sir. I'm Shashwat. Sir, I was
Sir,
thinking
sir.
if these type of sensors can be printed upon any biopolymer, something like that, so
Yes.
that we can use it for real-time monitoring of packaged milk.
Yes. Yeah, that's why I see. I just downloaded this paper, right? Just so we can print something like this on a flexible substrate, like this. You can see.
Yes, sir.
Yeah, you can definitely print on a biopolymer, something like this. Yeah, that's I'm just giving you. Yeah, now I'm giving you the idea, and you can use it anywhere, like in a paper, on a like a plastic membrane, or in a flexible substrate, and you can use for some different purpose. Hmm.
Yes.
Okay.
Yes.
I will recommend you just after getting all this your knowledge, please search some based on some keyword on the Google. Just I think I already many times I taught you how to search some just keyword. You will get lots of publication, and I will say no need to read the full paper. Just check it. Read the abstract, read the conclusion, and see the schematic, and leave it. Just check. So if you sit just 1 hour, maybe you can get almost maybe 20 to 30 ideas within an hour. You will get idea, right? 20 30 idea you will get. Then you can think and sit just silently sit and think. Let's try to coordinate all the idea and get some and like last class I taught you how to write the proposal. So you can think about let's try some new one. I can write a proposal. Okay. Okay, sir.
Yes, sir. Thank you, sir.
It's very interesting. Okay. Anuprabha Chatterjee.
Sir.
Yeah.
Hello.
So is it clear to you?
Sir.
Yes, tell me. Yes, tell me. I can hear you. Hello, Anuprabha? I think you're silent.
Hello.
Yes, hello. Bhavesh?
Yes, sir. Hello.
Yeah. Yeah. Is everything clear? Okay.
Hello.
Yes. Tell me.
Sir, like, can we prepare something related to with the respirated gases from the plant? Like, if we say ethylene, which is generally used, means uh, important in the ripening of fruits.
Very good question. See, that's something. There is so many area to explore. I'm just giving you a very generic idea. So this is called the gas sensor. If you like from your breath, right? If you want to detect something that come from your lungs, but some gas sensor you want to develop, right? This is your point. So you can search like gas sensor, or that is called gas sensor, or you can say gas biosensor, or like you can say, or you can say breath sensor also. You can get like breath biosensing. See, breath biosensing using electrochemical sensor. So I can download this.
Sir,
Yes, tell me.
as Bhavesh asked, like he asked about ethylene, right? So will it come under
Yes, sir.
biosensor or another like?
Your voice not clear. Please repeat.
No, sir. Like
Sir,
Bhavesh
I
asked regarding the ethylene, right?
Hmm.
I have recently studied about a sticker like which is placed on fruits, and it get
Hmm.
activated by the ethylene. Like when the fruit ripens, it releases ethylene,
Yes.
and the sticker turns into
Yes.
green color, like stating that it's
Yes.
ready to eat. So will it come under biosensor or it is another different like, type of sensor? Like it's not bio-related, right?
See, generally, see, we are. What is the biosensor?
asked.
So biosensor means, so we are sensing some biological molecule by using some receptor, something like this, or using some maybe it can be the nanomaterial, something like this. So it is all are the sensor. It is, you can say it is the biosensor.
Okay, sir. Thank you, sir.
You can say it is the biosensor device. See, here you can see one paper. Here also I am showing you one paper, like breathe biosensing. See, here is saying breathe biosensing, because from the breathe. Also, sometimes that for like security purpose, not only security, the traffic police, they want to
Drunk
take.
and drive test.
Yes, the driver, I mean, the drinking and driving, that is prohibited, right? But still, the drivers are drinking and driving. So how? So they are using some gas sensor like this. So they can hold this one in front of your mouth, and they can detect the level of the alcohol. So you can see this paper, breathe biosensing. So your sensor will be immobilized with some enzyme or maybe some nanomaterial. So when you breathe, I mean, gas can be captured on the surface, and it can show basically some color change. Basically color change. Not only the color, sometimes maybe color very less intense. We cannot differentiate the change. So in that case, you can use the electrochemical technique. Maybe by using your smartphone, where you can apply some potential and how much gas present on the surface that we can detect. Because gas can be absorbed on the surface and we can detect. The same technology you can try. So this kind of paper you can maybe follow. Okay, clear? Very good question. Any more questions from your side? Vibhuti, do you have any question?
No, sir. Fine.
Very good. Bhupendra? Hi, Bhupendra. Bhupendra Patel.
Yes, sir, I am listening.
You're listening, right? Everything clear?
Not at all clear everything. I have to study again.
Yeah, definitely.
Yes,
Because
sir.
I'm giving you all the idea and going to clarify you. But if something is not clear, you can ask the next class. And if right now, if something you is thinking that is not clear, you can ask me. Vibhuti.
Yes, sir. Yes, sir.
So which part is not clear? You can tell me.
Sir, actually, in the slide before this
Which one?
E. coli test after that thing. Yes, this
Your
slide.
voice is not clear.
Hello, sir. This slide only. This slide in the left side.
This slide, milk quality.
Ye- Yes, sir. Yes, sir. This slide
Yeah.
only.
Okay. Yes. Yeah. It is. It is. I mean, it is like the calibration curve that we got from here. This one. I mean, yeah. If you say read one, yeah, I will definitely say just read one time in your home and try
Yes.
to understand again, because I'm just giving you this is the calibration curve. See, like the small this is the small piece of paper, right? This is the small piece of paper. And in this area, you can see all the paper we put here, small circle. See, if you increase the ALP concentration, you can see the intensity increase, the color change. You can see the color intensity increase, right?
Yes,
That,
sir.
see the signal also, that also increase. That's
Yes.
that signal also increase. That signal actually we are getting from this mobile phone. We just from the nectar you can see the actual color change, but by the mobile phone if you use and you capture the color and you can analyze the color by using the algorithm, by using the app, so you will be more accurate change you can see. So that accurate change actually is shown in the left hand side. This is nothing but just a calibration.
Yes, sir.
This RGB means the red, green, blue. That the color. That color means that come from the. No need to get the very deep knowledge from here, but it is like this, like this output, and this is the concentration.
Yes, sir.
Clear now, Bhuti?
Sir, Bhupendra, sir.
Sorry, Bhupendra Pai.
Yes, sir. Yes, sir.
Good. Thank you. Anyone else? Harshita, is it fine? Everything?
Yes, sir.
But
We have to search some research papers and come up with some ideas.
Sorry.
So we need to search some research papers so that we will find
Yes.
some idea like to work
Yes.
on.
I'm just giving you the idea, everything. Yeah, because now, because the main fundamental things I taught, now I'm giving the example, the application side. You just search it and you will get more and more application. Because within the class time, I cannot show you all the application. Just I'm giving you some application. You can try searching like this way by using this kind of keywords in Google, right?
Yes, sir.
Okay. And Harshit Chaudhary, Harshit, are you there?
Yes, sir.
Yeah.
Hello.
So are you getting the main principle of this sensor, right?
Yes, sir. Yes, sir.
Very good. Very good. If you don't understand anyone, so please ask me right now. I can clarify you. Mohit Kumar. Hi, Mohit. Mohit. Mohit, you are not there. Okay. Rinchen Narbo.
Yes, sir.
Hello, Rinchen. Hello. Hello.
Yes, sir. Yes, sir.
Okay. So how are you, Rinchen?
I'm good, sir.
Okay. So are you getting the idea of this sensing technology?
Yes, sir.
Okay. Okay. Good.
Okay.
Shomo Vishwas.
Yes, sir, more or less okay with the idea.
Okay. So yeah. So this is the things for say another technology that is called the Okay. So again, so let's. See another technology. This is the magnet. I mean, we can use the magnet for the biosensor development. See how we can use the. This is called the magnetic. I mean, like a sample. Just I'm teaching you step by step. So left hand side here, like a sample, it contains your target. So you can synthesize some nanomaterial that has the magnetic character. Okay, magnetic character like the ferrous like Fe3O4, like iron base nanomaterial. So iron based nanomaterial on that, like I'm showing the cursor on the iron based nanomaterial on the surface, you can immobilize your secondary antibody, the antibody 2, secondary antibody, the detection antibody. You can immobilize. Now that nanomaterial can behave as a like a tag, or this is kind of labeling, right? Like here, like the secondary antibody that conjugate to the ALP, right? That secondary antibody you can conjugate with ferric oxide, like Fe3O4. So Fe3O4 ferric oxide nanomaterial you can synthesize and you can conjugate with the antibody. Okay, see this antibody conjugated these see iron oxide, they have the magnetic property. Now you can drop this magnetic behavior as nanomaterial that already conjugated antibody, you can drop inside the sample, like the water sample that contain E. coli as E. coli bacteria. So your antibody that conjugated your nanomaterial that is specific for your E. coli. So they will bind with the E. coli, right? See now you can see this chamber. There is the E. coli also. There is some other interfering, other biological material also there. So magnet actually basically help you to wash out other thing. Only your target one will be there. Other thing you can wash out easily. So if you hold a magnet here, see what will happen. This iron nanomaterial can hold the E. coli. So if it conjugates with E. coli antibody, hold the E. coli and other bacteria, other independent species, they will be the solution. Now you can decant and you can easily wash out all the other material that you don't want. Clear? Right?
Yes,
Guys?
sir.
You
Yes,
can do
sir.
it, right?
Yes,
It's
sir.
very beautiful technique. You just hold the magnet. They will just touch. I mean, they will be one side, and other species will be the another side. So you can decant, you can remove. See this number third of this chamber. You only have the E. coli bacteria. You don't have any other thing, right? So now you can apply the same thing like these things. So you can develop a sensor surface so that contain your primary antibody. So now the solution that contain your secondary antibody and E. coli you can drop and they will bind on your surface. Now you can try this chronoamperometric technique or cyclic voltammetry and whatever you want you can try. So this thing I can try. So on the surface, this is kind of like your electrode. So on the electrode surface, you can immobilize your primary antibody. So that primary now if you like this, you can see my cursor. Like if this sample you can drop on your sensor surface, so they will bind. So basically, you are avoiding, I mean, you are eliminating all the interfering species by using a magnet. So you can hold a magnet maybe like this way, or maybe you can hold a magnet like a magnetic surface. You can try on that magnetic surface. Maybe you can hold the magnet on the bottom of the hair, and they will bind. And you can wash out by using some buffer sample, or you can use like similar this kind of technology because all the intermediate species will be washed out from your sample. Then you will get only E. coli. This is the main principle here why you are using magnet. Yeah, guys. Do you need any clarification? Hello?
No, sir.
Yeah. Yeah, just understand the basic principle. So if something, if you say, if you don't understand, maybe very deeply, so it is fine. But just understand the basic principle. Then you can use this knowledge for your practical application. Okay. Okay, guys. Fine.
Yes, sir.
Okay.
Okay.
Yes, sir.
Very good. I think that's all for today. Then let's take your attendance.